Good morning, everybody. Thank you for coming, and thank you, Ruben, for having me. Thank you, Marilu, for organizing this amazing thing. And it's a huge pleasure for me and honor to give the first talk here in front of all those people who know a lot about the system and the threat that we are all facing now. So I hope everybody is satisfied. If not, I would love to hear your opinions and comments and questions after my talk. So what's wrong with ambrosia beetles these days? Well, I'll divide my talk into three sections. One is, we do know something about ambrosia beetles already as a group. Then there are things that we think we know, but we may need to change our view, just like Ruben emphasized. And then there are things that we just have no idea. Some things are new. We have never seen some of these phenomena before. So how do we deal with that? I will start with what we all know. We all know that ambrosia beetles are fungus farmers. We know that they live inside wood, in galleries or tunnels like that. This white stuff on the wall of the gallery is the fungus that the beetle feeds on, that the beetle brought into the gallery and essentially planted there. These are eggs. This is the new generation that will hatch and eat the fungus. The beetles don't eat wood, they eat the fungus. We know that the beetles have mycangia. Mycangium is a fungus pocket. It's a pocket inside the body of the beetle. In Eulacea fornicatus, this is probably what we'll see. I'm not entirely sure that this is what it looks like. It's a related species. But we see that if you slice the head of the beetle like that, this is the head. It's about 0.5 millimeter big, but it's very small. But if you slice it, you see, these are the eyes, this is the front of the head, and these two pockets up front here are full of something. The something is the fungus that gets introduced into the galleries. We know that many ambrosia beetles, particularly the group in which Eulacea fornicatus belong to, are haplodiploid and inbred. What does that mean? That means that the females are all just like regular beetles, like other animals, like elephants and pigeons. They are diploid, meaning that they have two sets of chromosomes. The males, on the other hand, have only one. They're mothers. They're haploid. Moreover, they're inbred. That means that the males, they never go outside of the gallery, or very rarely, sometimes they do, but rarely, they mate with their sisters. There are a whole bunch of sisters or daughters in each gallery and only one or very few males. The males mate with the sisters and die there. So it's pretty much only the females that you ever see outside of the gallery, and they that cause any damage. We know that ambrosia beetles are actually a hugely diverse group of organisms. There are about 3,500 species, almost all of them in the tropics, but many of them are being introduced. This is a kind of a cartoon of a classification of bark and ambrosia beetles. We know that the ambrosia symbiosis evolved many times. So ambrosia beetles are not a single group. Many different bark beetles evolved the capacity to live with the fungus in a symbiotic habit. And the Eulacea fornicatus belongs into one of those groups, the Xyloborini up here. But there are many other groups that converged, evolved the same strategy. And we know that most of these beetles are harmless. Literature these days would try to tell you otherwise. It seems like that the publications that are now published have been in the past 10 or 12 years. Most of them are on pests or invasive ambrosia beetles. It seems like most of them are bad. It turns out that if you relabel the axes and if you put species diversity, it looks like this. Vast majority of those beetles are deadwood colonizers in their native habitats, and only very few species are now emerging as pests. Unfortunately, this minority are emerging as major pests. And that brings us to the new chapter of my talk, which is what we think we know about some of these pests, what we are starting to see. And I'm going to talk about, in this case, I'm going to mostly talk about, not about the whole group, but about the Eulacea fornicatus, the beetle that we have here with the fungus that came with the beetle. And I'm going to talk about what we think we know about the beetle identity and about the fungus identity. Now, I have not studied this species very deeply. There are people in the audience who know much more about it, and so again, I would love to hear your feedback. First, the beetle identity. Normally, if you have a different kind of species of animal, like a pigeon, you would put a name on it and you know what it is. You know that the name predicts something about the organism. So you would think that initially when this beetle arrived here, we thought that, well, it looks like Euvolesia fornicatus, it's probably going to be that, so we'll put the name on it. And it'll probably behave like Euvolesia fornicatus normally does, which is not the case. We know that this beetle that we call Euvolesia fornicatus is causing major damage in Israel and now in the United States. They're attacking live trees. Now the problem is that an organism with the same name is causing damage in tea plantations in India, but a completely different damage. The beetles are boring into twigs, the twigs snap, and that's the damage. It's not a virulent pathogen in any way. It's really a mechanical damage. Now, I studied a lot of these insects in Borneo and Papua New Guinea and other places in Southeast Asia, and it turns out that there, an organism with the same name is boring holes into branches of Artocarpus and Ficus, family Moraceae, a completely different ecology again. So what's in this name, really? It turns out that a lot of these Xyloborini, these haplodiploid and inbred ambrosia beetles, are not well-defined species. They are a patchwork of clones because they reproduce kind of clonally. They're a patchwork of different populations that sometimes have local adaptations that are different than in other places. And we are starting to see that this might be the case with Euvolesia fornicatus. Why does that happen? Here's a small cartoon of how a population of a regular species, any other beetle that's not haplodiploid and not inbred, develops through time. We see that these are individuals and they go through life and they meet each other and mate and produce families and the whole population goes up. And so if there's a mutation, let's say you have yellow dots happen to come up on a beetle, there's a good chance that the mutation will spread across the population. This is how species maintain their identity, because genes can travel through the population over time. It's different with haplodiploid and inbred species, or I should say populations. They very rarely cross paths. It's mostly, sometimes it happens, but it's very rare. And so if there's a mutation in one of those clones on some island somewhere, it very rarely, the gene rarely makes it into any other population. And so what we see are these, again, patches of clones or populations in the world that are diverging constantly and becoming more and more different. And it's really difficult to put a species name on those populations. What does that do to the DNA sequence of these insects? Well, here's an example. This is from my work in a previous lab. This is several species of ambrosia beetles, very closely related. And this is their 28S ribosomal DNA. And we see that some of them have this big chunk in the middle of this normally relatively conserved gene. It turns out to be probably a microsatellite. For those who don't know, I'll be happy to talk about that later. But the point is that they have sometimes very, very divergent or different sequences of DNA, which we are now trying to use to identify the species. And so what happens with that in populations that are living in different parts of the world of what we call the same species is this. Here's another species of an ambrosia beetle. This is one I studied with my colleagues in the previous lab. And this is a divergence. This is a percentage difference in a gene called the 28S that we saw before on the picture. And here's the cytochrome oxidase 1, which is often used for identification, molecular identification of species these days. And we see that there are very big differences between different regions of the world. These numbers, this is 10% of bases of nucleotides in their gene is different between different populations. This corresponds to the level of genus in other animals, hugely divergent, and it still looks exactly the same. And so even using molecular data for distinguishing these species is not easy. It doesn't give us all the answer right away, and so as we talked about that with some of us yesterday, it's not as easy to say this is the same or this is a different species, mostly because we don't really quite know what a species is in these clonal populations. And that's a food for more discussion, I hope, later. And we'll also probably hear other people talking about the morphological diversity, which I'm not going to talk about. Fungus identity. What do we think we know about the fungus? And again, I'm not a mycologist. There are people who know more about that. So I would love for this to be food for thought and for more discussion. My question to all of us is, this is what was called Fusarium ambrosium in 1947 in a paper that described the fungus, the symbiotic fungus of Uvulaceae fornicatus from India. This is what it apparently looked like in the gallery 60 years ago in a completely different environment and different continent. Is it the same that we have here in California or in Israel, in Australia? I don't know, and I'm hoping that people will tell me whether or not it's the same. There is one little problem. We know that ambrosia beetles do swap or change associations with their fungi. Here's an example. I did study a few specimens of Uvulaceae fornicatus from Papua New Guinea, and I saw none of Fusarium. I saw that 100% of the galleries had something related to Ceratocystis. It was not Ceratocystis, it's an unknown fungus closely related to Ceratocystis. So, no Fusarium. How is that possible? The other question, the other thing that we think we know is that we think that the Fusarium, or the symbiont of the beetle, gets transported in the mycangium, which is the fungus pocket, which in this species, Uvulaceae fornicatus, is supposed to be in its mouth. I believe that's where it is. I haven't studied this one in particular. So, which would be kind of like here in front of its head. Now, we also know that ambrosia beetles carry lots of other fungi, some symbiotic, some not, on different parts or in different parts of their bodies. In the mouth, they have all kinds of stuff in their mouth when they're flying around. They have lots of stuff, spores or hyphae on their surface, and especially underneath the elytra. This is just a modified wing of a beetle, really. This whole part of the body is hollow. They can open it up, and that's where underneath, that's where the actual wings are. And that's a hidden ecosystem. They have mites and nematodes and fungi there. I don't think anybody has studied that. It's possible that they have lots of different fungi there. How about in their gut? We know that there are lots of fungi transported in the gut of these beetles. So I think rather than just taking a beetle, squashing the whole thing, and isolating whatever grows from it, it would be better to understand where and how these associates, the fungal associates, live. It would be better just really to take greater care and look at individual parts of the beetle. The reason why I'm saying that is that there are data from other systems which warrant more care. One is that in Xylosandrus germanus, which is another ambrosia beetle that's now spreading in the Midwest and starting to cause damage in nurseries and even in the nature. In 1947, there was a report that the beetle is associated with another Fusarium, which causes cankers on walnuts in nurseries. So when I find the beetle, I open up the twig in which the beetle lives, and these are the pupae and larvae of the beetle. This is the cave or the gallery in which they live. And I take a sample from the gallery, and indeed, what I see is Fusarium. Almost pure culture, beautiful pink Fusarium. Now the problem is that when I take the beetle itself, it looks like Uvulaceae fornicatus. It's not even related. The mycangium is in the middle of the body. It looks like this funny shaped organ. That's where they carry the fungus. When I take that and plate it on a petri dish, I get a pure culture of Ambrosiella. So there's a difference between fungi that they carry in their body, in their mycangium, and on their surface, and the fungi that we get growing in the gallery. So we think we know that there's this close symbiosis between the beetle Uvulaceae fornicatus and Fusarium ambrosium. But I think there's still a lot to be learned about that. And so I have a student who is actually going to look into that in the upcoming semester, looking at individual parts of the beetle and what fungi are carried in different parts of the beetle. Now, the last part of my talk is going to be about things that I don't think anybody knows for absolutely sure. I certainly don't. And so these are, consider them questions, consider them observations of one guy who happened to see this, who happened to study these beetles for a few years, but really have seen the actual effect of this species just for one day yesterday. We've been debating whether or not these are primary or secondary pests, meaning whether they can kill trees just indiscriminately or if they just go after trees that are somehow predisposed or have been stressed or something like that, which is what ambrosia beetles often do. We don't know what the disease is going to be like in the future. We don't know of the behavior of this, if you want to call it a disease, these symptoms, this complex, going to be in the future. And we are just now starting to think about what to do about this. So first, let's talk about, is it a primary or secondary pest? There, according to my many discussions with many of you yesterday and the days before, there seem to be a range of opinions that I think can be characterized by 2 points, 2 opinions. One is that the beetle and fungus are indiscriminate tree killers and they kill everything they meet. That's one end of the spectrum. The other end of the spectrum that I hear from other colleagues is that the beetle and fungus seem to be only going to stress trees, trees that have somehow been predisposed for this attack by other fungus, by drought or overwatering, whatever. So let's look at some pictures, some trees that we've seen yesterday. This is the, I believe this was box elder, I'm not quite sure, but we've seen that this is a dead tree. There's no question about that. All these holes, 1, 2, 3, 4, 5, you see the beetle here, 6, 7, many holes. This is a major infestation. The tree would be very happy living without the beetle around. There's no question about that. The beetles kill the tree. You would think bad primary pests. Now, we also saw pictures like this. This is an ancient avocado tree. I hear from Tim that we don't even know how old this is. And here are these, what are called the sugar volcanoes, the attempts at attack by multiple beetles. Well, we see that there is something wrong with the branch. I believe it was cut here or here or something, and it's very localized. The whole rest of the tree seems to be healthy and not attacked. So when somebody else looks at this kind of situation, you would think they're just secondary pests. They're not even pests because they're not even reproducing in this. We actually managed to scrape some of these volcanoes on other trees where we could reach, and there were no beetles. This is just an attempt, an attack that the tree was able to defend against. We also saw cases like this. This is a beautiful olive tree, again very old, and we see these little, what was holes, probably the beetle trying to somehow randomly landing and tasting and maybe inoculating the fungus, and the tree is defending itself, oozing the sap and resin, and seems to be doing quite fine. It's not really dead. The problem is that, you know, okay, so we see local damage. You would think that's a secondary pest. We see beetles landing seemingly randomly on completely random selection of hosts, like an olive tree, and trying to eat it, but not really succeeding. But then we also see this. Trees that are completely riddled with beetles and definitely dead. I think we are looking at different stages of the behavior of this problem, of the complex. It indeed probably most often starts as either local damage, something that the ambrosia beetles just do normally. They go after weak branches or weak trees. Sometimes the tree defends itself, sometimes it doesn't. We see that the beetles are also trying to taste trees that kind of look, smell tasty. We know that a lot of these trees that have lots of nibbles on them have been heavily watered, and we know that ambrosia beetles do smell these trees, and they like it because these trees, the overwatered trees, emit ethanol, and so we know that the beetles like that. The problem is that if you have too many nibblers, it ends up like that. And then the second problem is if it ends up like that and you have too many beetles being produced in a locality, those attempts are increasing, and then there's a point in which there are too many nibbles on a tree and the tree can defend itself. So there is a feedback. We are looking at different stages of the infestation in the landscape, I believe. And I would love your feedback. The thing is that this is not new. This is exactly how many other beetles, bark beetles and ambrosia beetles, behave. The most textbook-like example, the southern pine beetle, or Dendroctonus frontalis, that attacks pines in the southeast, in Texas, in Mexico. Normally, in the vast majority of cases, the beetles try to drill holes in a pine, and the pine is defending itself, it's oozing sap and is pushing the beetles out with resin. And in most cases, the beetles just taste it and are killed, and nothing really happens. So you would say in most cases, they only go after diseased or weakened trees. That's absolutely true. The problem is that when they succeed or when the tree really is weak and the beetles succeed in overcoming its defenses and breed successfully and build up a population, this is what you see. Massive outbreaks. These beetles are capable of killing everything in their surrounding if there are enough of them. So suddenly you see the progression from what is normally a secondary pest into a major outbreak on a vast scales, and clearly they are primary pests. So by labeling something primary or secondary, it's not really a label of a species. It's a label for a population that's in a certain stage of its development. So I would say that it's both. It's both primary and secondary pests, and it depends on how many are flying around, how weak our trees are, and if we do something about it. I think this is a very important point that I will talk about a little bit later. This brings us to the discussion of the disease and its future. Now, before we discuss that, we really need to understand, I think, the mechanism of the disease a little bit better. And let's say there is this gradient of virulence of fungi associated with beetles, bark beetles and ambrosia beetles. On one end, here's the laurel wilt. This is a disease that's now being spread in the southeast by the red bay ambrosia beetle. Many of you have heard of that. The fungus is super virulent. One beetle inoculation can kill a whole tree in just a few weeks, whole giant avocado tree, just a few weeks. Super virulent disease. On the other hand, there are many bark beetles that are capable of killing trees, but they're doing it by themselves, by swarming on a tree. The fungi are somehow there, but they're not really important. The fungi are not virulent. And then there are cases in the middle. And this is an example of the recently spreading thousand cankers disease of walnuts. You need both the fungus and the beetle. The fungus is somehow, I wouldn't call it virulent. It's pathogenic, but locally. But if you have enough of those, if you have enough of those nibbling beetles, you do get the death of a tree, and if you get enough trees, you get the death of whole populations of trees. And my gut feeling right now, after seeing the situation yesterday, is that Uwalacia fornicatus and Fusarium ambrosium are somewhere there, somewhere in the middle. You need both. The fungus itself is not a very good pathogen. It's not very virulent. It's mostly localized. But then if you have many inoculations, then you get a problem. So this is essentially repeating what I just said. The fungus itself may not be super pathogenic, super virulent pathogen, but if you have enough of them, like in the case of the thousand cankers disease, no one has heard of that 10 years ago. Now this thing is spread across the whole continent, and it's threatening walnuts with extinction. So again, we don't need a major pathogen. But if the beetle helps it, and if there are a lot of beetles, and there are, we end up with a major disease. So there are good news and bad news. I think the bad news really is that this beetle kills trees that otherwise would be perfectly happy. Some of them may have been overwatered, but they would do well. They grow just fine without the beetle. The beetle kills them. That's sad. The dead wood is super contagious. One tree can, if it's heavily attacked, and they often get attacked after they die, they produce thousands and thousands of beetles. Very, very big problem. And if we do not do anything about it, we are up for a major problem. This is my gut feeling, that if we don't do anything at this point, we may be past this point, I don't know, but I still think that there is hope. If you don't do anything, the populations are going to build up, and even if we now can think it's a secondary pest, it will become a primary pest, and we are seeing this in some trees around here. The good news is that I'm still an optimist, and I think there is something that can be done, at least locally. I think the major explosion may be preventable, or at least local explosions may be preventable. We see that the beetles they don't really belong to this environment. And so we've looked yesterday at every single dead tree that I could find. There is not a single beetle in dead trees, which is their normal environment. So if the beetles run out of live hosts, I don't think they will stick around for much longer. I mean, it's not a very good news, but there is no really a long-term reservoir for those beetles. And the other good news is that even if we see attempts at initiating a gallery, it's not always successful. In fact, majority of the trees we've seen yesterday do not produce beetles. So I would not call those hosts. Most of the trees we've seen suffered a little bit of a damage, but they are not hosts. They don't produce the vector, and they don't help the disease in spreading. It's only when they really go past the point of no return. So what to do now about that? It's clear that we need to do something. There's no question. If we don't do anything, we'll end up with something like the southern pine beetle only across whole California, and who knows what's going to be that. I think we can start with solving fairly basic problems. Sometimes, we know a lot about other beetles, so we can take notice. We can get inspiration from other systems. First of all, I think it's important that people know about this. Growers need to know about this. The public, the people who make decisions about city trees, about watering, about everything. We really need to make noise. Very importantly, the wood really is contagious. We know that, and we know that about other systems where other diseases that are carried around by beetles. We know that there are campaigns against moving firewood around, for example, in other states where they have other beetle problems. These campaigns work. They don't solve the problem, but they slow the spread, and that's very important. We need to start, I think, studying some of the relatively basic ecological features of this new disease or this new pest. We need to develop attractants. Maybe they are simple. Maybe it's just ethanol with something. Maybe they are simple, and maybe they will help us again. They will not solve the problem, but we know that even with a very finicky pest like the red bay ambrosia beetle in the east, there has been progress made, and we are figuring out slowly what the beetles like, what they're attracted to, and maybe we'll develop a trap. And I bet you that Eualacia fornicatus is going to be easier because it's not host-specific. So it's probably going to go after something very generic. I think we really have to focus on the wood that's producing beetles. The chunks of trees that, we don't have to treat every single beetle that has a little lesion, every single tree that has a little lesion. But the wood that's breeding the beetles is contagious, and we need to start figuring out what to do with it fast. Is it possible to just let it dry out? Is it possible to chip it? Do the beetles survive it? Is it possible to just throw a tarp over it and bake them inside under the California sun? Or can we use it as a temporary trap? Because those trees are very attractive to more beetles. And we've seen that yesterday, that if you have a tree that's freshly dead, it's super yummy for many beetles. And so we can use those trees to trap out more beetles and then chip them or something. Get rid of them really fast. And we need to start figuring out if new practices in horticulture, in silviculture, in arboriculture, in avocado plantations and everywhere will help this. Maybe we just need to look a little more carefully about the watering regime, because it turns out that a lot of those trees we've seen yesterday heavily attacked were heavily watered, or were too dry. I don't know, but that's what I think we need to start looking into. Some basic answers. What to do with these attacked hosts, how to prune the attacked branches and how to get rid of them. Maybe we can just do preventative trapping. With lots of traps in an area, if it's attractive enough, you can bring down the population to the point where there are no longer primary pests. And we know that injections of fungicides work in other systems, and they seem to be working in the system too. It's expensive, but with valuable trees, it's possible, again, to bring down the population, at least locally. So I think there is hope, but we need to do something. There's no question about it. So as a conclusion, the species identity, it's an exciting question. It's not like identifying a species in many other groups. We see that this is not a species like many others. We see that it's a global mosaic of different strategies, different DNA sequences, different morphotypes. And I'm looking forward to a definitive answer of what is Fusarium ambrosium, and if it really is the symbiont and the tree killer. We are starting to see that the disease dynamics is kind of emerging in front of us. So we are starting to see that if we do nothing, we may be up for a big problem. And also the other thing is, I think we need to start paying attention to this whole complex, not just to a beetle from an entomologist's perspective, or just to a fungus from a plant pathologist's perspective. The disease or the pathogen or the pest work only as two together. So it makes really no sense to try to discuss the disease only from the fungus perspective. And the definition of host too. I don't think that all those trees are a host. They are potential hosts, but only really at certain level of the infestation. They can really produce beetles. So again, it's about the level of the density of beetles. And what to do now? Well, there has been some work done in other systems, so we need to look at that. We need to start exploring simple things and affordable approaches to getting rid of these beetles, at least from important areas like avocado groves or botanical gardens or cities. And we really need to make noise, need to tell everybody that this is a problem, and if it's not a big problem now, it will be if we don't do anything. So thank you very much. And thank you, Ruben, for organizing this whole thing, and Mary Lou. And again, this was a one-man perspective, so I would love your criticism and questions.
So, you know, it seems that from a one-day visit, you did learn a lot of things, and so I do wonder. It's of course just a snapshot in time of what you've been seeing. And for instance, you mentioned the avocado tree there, that it's just at one branch and it's really no big deal. Obviously, in Israel, they have different perspective on that. And it may simply be a matter of time. And,
absolutely.
So it seems to me that, you know, we are in a really early phase here, and trees not dying immediately is obvious. It's early in the infestation, and I think, you know, and your remark about getting rid of the trees that do produce offspring, you know, in let's say in the greater LA area, they like to breed in Quercus agrifolia that is planted along many a street, and so getting rid of those trees, and it's a native tree, also maybe you know, just beyond possibilities in the short term. So I think we do have, like you say, a major problem, and I don't think that we can simply solve things by removing the host.
So I perfectly agree with the beginning of your statement, absolutely 100%. We are at the early stages, and if we want to prevent an outbreak like the one we see in Israel, we need to do something. I agree with that. Maybe I misspoke about removal of hosts that produce beetles. I did not mean the whole species removal. What I meant was that if you remove 90% of the trees that are now trees individuals that are now producing the beetles, you would knock down the population significantly, and it may help. Yeah. And so I'm not proposing, you know, clearing the streets of trees. What I'm proposing is to let people know that we have a problem, let homeowners, garden owners, nursery owners know that if they see something like that, it needs to be disposed of right now. Yeah. And not the whole species, but the actual trees that are teeming with beetles.
I agree. And the unfortunate thing is, like, we've been talking to the nursery industry, and the nursery... Oh, thank you. See, is this thing on? It should be on. Okay, so the nursery industry, you know, I'm sure they want to stop this thing from spreading, but we don't have anything to tell them what they can do to their living trees. Okay, you got a living tree, it's infested. I'm a nurseryman, I want to sell the bloody thing, and I can only sell it if it's free of this beetle. What am I going to be doing? And we cannot tell them, oh, you know, you just dump this stuff on this plant,
and the beetle dies, and you can sell this healthy tree. So it's, you know, I think a bit of a problem that we have at the moment is that we're indeed so early on in the infestation, and we can come up with you know, wonderful, you know, let's say theoretical solutions. Unfortunately, the reality of the situation is such that, first of all, there's a huge area already infested, and at this point, we don't have anything to offer somebody who's got an infested tree, except for saying, get the thing, kill the tree, and then we don't even know how to dispose of the wood properly. So it's a problem.
It's a matter of research that needs to be done right now.
Yeah, Jiri, just to me, one of the complicating factors here is, again, this complication that there might be cryptic speciation within the beetle and the fungus. But that said, it seems to me now that this thing is being seen in a lot of different environments. I'm from Florida. We've got people from Australia here. We've got people from Israel and California here. There's an opportunity here, I think, to understand what these various factors are that do result in major damage. What distinguishes Israel and California from Florida, for instance? Understanding that then might, in various locations, and we could address whether overwatering or these other factors play a role if we understand in a geographical sense what's going on in these different countries. So, nice talk.
If I may, just a quick comment on that. I absolutely agree. I think it's not a matter of an insect and fungus. It's a matter of an insect and fungus and the landscape and hosts. And yes, as a sort of more of a theoretical hunt for questions, this is the first one that I agree should be solved to be even able to predict where else it's going to go next. But the sad case is that I think we should get on it locally and figure out what to do with those damn attacked trees. Yeah.
Jiri, just wondering, I guess the obvious biological question is about inbreeding depression of the species, and also I was just thinking you could imagine in a wild ecosystem where you had like a population of clones and more than one clone could colonize a tree. Is there any chance of sort of crossover between brood galleries of males?
Right, very good question. It has been studied a little bit. There are very few papers on it, but they exist. And we see that even though the vast majority of the families never interbreed with any other family, the males always stay in their gallery and just die there after mating with their sisters, there are occasional males that come out and crawl on their tree. They are flightless. They're like wimpy males. They really don't have wings or anything. They crawl on the tree, and if they find other gallery, they try to get in and mate with other sisters. So there is an occasional outcrossing in those normally inbred populations that is responsible for a little bit of a gene flow. It's nothing compared to irregular outcrossing populations, but there is a little bit of that. So they're not entirely clonal. They're just mostly clonal. And I'm hoping that answers your question.
It does.
Okay.
Jiri, oh, thank you. Very nice talk. Thank you. I enjoyed it a lot. I would like your thoughts. I know this will be pure speculation, but given what you know on ambrosia beetles and these other invasives, the 13 that have moved globally and caused big problems, based on what you know about those systems and this, do you have a feeling for how this beetle may have come to California? Do you think it was moved on live plants through the nursery trade, something like that, through packing pallets? And based on what you know about the phylobiogeography of these things, do you have an inkling as to perhaps what parts of the world this beetle may have originated from, and whether there were trade pathways that could have facilitated that movement?
Yeah, excellent question again. Fortunately, there has been research on that already, at least theoretical research, quantifying the methods or the pathways how these nasty invasives get around the globe. And in terms of wood-boring beetles, it's very often wood pallets, for example, packing material, solid wood packing material, untreated wood that just gets shipped without any control around the globe. And if you ever have a chance to witness what happens when a giant container ship arrives to an LA port or somewhere. And when they open up those containers, look at what's coming out. There are so many insects that I'm surprised that we don't have many more problems. It's pretty disturbing. I also hear, I'm not sure if that's confirmed, but I also hear that the ports of entry facilities have only enough manpower to check about 2% of cargo, which is nothing. It's not even monitoring. So wood is a major problem. Live plants, soil, another major problem. These things can survive for a long time in wood. So my gut feeling would be that wood is most likely to be the culprit here. But you know, any number of other ways how these things could get around. The problem is that there are regulations for treating wood, treating packing material before export and import into other countries or continents. But they require investments, and not everybody does that. And that's a problem, mostly from the tropics, where most of these beetles are, and where the countries usually don't have money. They just don't do that, and that is a problem. So I have no clue where this species came from. I think Richard will give you a better answer, because I believe you guys are working on a population structure, global population structure of the species. So I don't know. I don't have any data to say, but so I don't know.
So just what you've suggested, that these beetles could possibly live in dry wood pellets. That's
I wouldn't say they would be happy there, but they can survive there, that's for sure. So if you have a teak tree somewhere in India and you make pallets out of it, when it's still fresh, the beetles will attack it. That happens all the time. They drill holes in it and they make galleries and they survive there if it's moist enough. And you know, if you are quick enough to use that pallet and bring it over to the United States, they will come out. Absolutely.
I would say on the pallets, it's less likely that they attack the pallets, rather that looking at some of those branches at the sites we looked at yesterday, I could easily conceive of cutting something like that up, cut it into pallets, nailing them, and it's the survival of those that are being carried along. So I think one of the things, and you referred to this on the trade and a number of containers, is just because you have an unlikely event, a low probability event, if you do it often enough. Yeah. So you know, it just takes one. So I mean, I don't think they're attacking the newly cut wood. That seems pretty unlikely, but certainly that they'd be in the wood that's cut up. So I think that's highly likely.
That is an
a
excellent
very good
point,
source.
that it only takes one. In case of these clonal inbred beetles, only one specimen is enough to establish a whole population, unlike in many other organisms which need a male and a female to mate. These guys don't. And so that's why, even though these beetles form about 2% of the global diversity of pests, they form about 50% of all invasive pests, because they're really good at invading.
Is there any idea of what food source is better, which fungi that is found on the different parts of the ambrosia beetle support that population better, and are those fungi better adapted to certain trees?
My observation is that, it's not hard data, it's my observation is that the fungus that is likely the best, most nutritious for them is the one that has co-evolved with them, and that has really co-evolved with their mycangium. There is some chemical communication going on in the mycangium between the beetle and the fungus. We don't know anything about that. But the beetle somehow makes sure, or maybe it's the fungus itself that makes sure that that one most nutritious fungus dominates in the mycangium. That said, we know that if you take a beetle out of a gallery and put it in another gallery that was artificially inoculated with a different fungus, the beetles do okay. So they are not entirely fungus specific. They're also slightly polyphagous in the terms of the fungus. And I always like to say that they're kind of like people. We don't just eat wheat or just rice. We have a certain number of plant species that we mostly eat, but we can really happily switch between them. So it's like the beetles. They have mostly one fungus at a certain locality, but they can swap it, and on the other side of the planet, they might as well have a different fungus, as long as it fulfills the feeding function.
I have some preliminary data on that, what we found on what part of the beetle and larvae, so I'm going to talk about it this afternoon.
Perfect.
So Jiri, I just had a quick question for you.
Oh.
So do you think that the feature of ambrosia beetles carrying virulent pathogens is a more sort of evolutionarily advanced feature? I mean, you know, most of them don't carry. I mean, they're normally going to trees that are already dead and dying. So why would they need to carry? Why would they carry pathogens with them if their normal sort of biology is that they don't need to kill the hosts? The hosts are already on their way out.
It's about the most complicated question you could have possibly asked. So very shortly, I will say that there's really, as far as I know, only one case where an ambrosia beetle carries a really virulent pathogen, which is the red bay ambrosia beetle in the east. All the other cases are cases where, or most of them are where you need many inoculations, many beetles to overcome a healthy tree. So they really don't, I wouldn't say that they carry virulent pathogens. That happens, but it's rare. What they often carry is fungi that are weakly pathogenic, because that's helpful. If your normal habit is to attack freshly dead trees, it's helpful to have a weak pathogen with you. Or I should say, for the fungus, it's helpful to be weakly pathogenic because they're better at conquering the surrounding wood or phloem or whatever you're up to conquering. And so they do have some capacity to overcome localized tree wood defenses. And so when you then get into an area where these trees have not been exposed to those fungi, those weak pathogens are become little stronger pathogens. And if there are a lot of them, we as people tend to see the end result. But the fungus, I wouldn't call the fungi virulent pathogens. That's rare.
Do we know much about the flight activity of these female beetles? Are they aggressive flyers, or do you see a spectrum from those that just crawl up the trunk to maybe species that can fly, you know, many kilometers?
Oh, so you're asking about the time of a day, or how far they can disperse?
Dispersal capacity.
Somebody may have studied that. If yes, let me know. What the little I know is about it, the curve. Here's a tree, and this is 10 kilometers of a distance. Large majority of the beetles will end up as close to the tree as possible. There are some that don't find anything, and they just keep flying until they find something. And so it's like a trailing end. So there is really no end limit that we know. They will just keep flying until they find something. And we know from, for example, Ips typographus, which is a European spruce beetle, they were found 60 kilometers away from the nearest attacked spruce. It's super rare, but it happens. But the vast majority of beetles, they don't have any interest in flying far away. They just have an interest to find a tree as fast as possible.
Well, that's assuming that dispersal only happens through flying. That's right.
Oh, absolutely.
A lot of movement around.
Absolutely. That is, by the way, the most dangerous thing, that people help them a lot.